Triacylglycerols, just like esters, can be hydrolyzed with water in the presence of acid, base, or enzymes. And in this process, we find our ester linkages and just cleave those and put water in its place instead. So the product of this reaction is our glycerol. And we'll get our 3 R groups, and they're all the same because we have tristerin here. So that's our product of hydrolysis, just like when we hydrolyze esters. We found our bond, inserted water in its place, and then we got the alcohol as a side product. So draw the products formed when given this triacylglycerol that is hydrolyzed with water in the presence of sulfuric acid. So find your ester linkage, carbon double bond to oxygen. This is going to get cleaved here. This will get cleaved here. So it's between the C double bond and the O that gets broken apart. So I'll get glycerol in the process. That's the first one. Second one. All right, and there is our 3 fatty acids that are products of our hydrolysis. When our body stores triacylglycerols, it gets stored in adipose tissue, which is made up of adipose cells. In adulthood, the number of the cells does not change, but the amount of stored lipid changes. Again, your number of cells does not, just how much your body is storing. So this acts as a layer of fat cells that gives us energy storage. And just below the surface of the skin, in your bone marrow, breast area, around the kidneys and pelvis is where it's stored. And that can provide us with 2 to 3 months of the body's energy. So we can see a close-up of adipose cells there. So that number doesn't change, but just how much is stored does change. If we have a typical triacylglycerol, this will get hydrolyzed and then metabolized, so it's going to get broken down further to form carbon dioxide plus water. And as a result of that, we also get energy, and that is going to be 9 kcals per gram. So if you ever notice, your fat content on different products are equal to 9 calories. If there's 4 grams of fat in a product, that's going to contribute to 36 grams of calories in whatever it is that you're eating. Adipose tissue insulates organisms and can be used for energy when it is metabolized. So if you have some triacylglycerol that reacts with water and lipase, lipase is an enzyme in our bodies, so we don't have to use sulfuric acid in order to hydrolyze these triacylglycerols. We find our ester linkages, these get broken up, and then we're going to get glycerol in the process. And we'll form 3 fatty acids. We have R prime, and the fatty acid that resulted from R double prime. So 3 fatty acids. These different triacylglycerols can be used for fuels, which you may have heard the term biodiesel, but this still gives off carbon dioxide, which contributes to climate change. So it is a nice alternative fuel, but not the best one. Soaps are metals of carboxylic acids, and you may recall discussing soaps in chapter 17. And this is an example of one soap, which is sodium stearate. And the way that soaps form is you take some fat or lipid, and that contains your triacylglycerol. This one happens to be trioluene. We're going to add 3 molecules of sodium hydroxide for every 1 fat. So this is going to undergo a saponification because it's under basic conditions. And then we're going to break up our ester linkages here, and we'll form glycerol. And then we're going to get 3 molecules of the sodium salt. So this is sodium oleate. which is a soap. And if you remember, the nonpolar long carbon chain tails are what dissolves the grease. The polar head of the carboxylate salt makes it water soluble. And different properties of soap, whether you have a hard bar of soap or the soft soap, the jelly-like stuff, that will depend on what fat source was used. So if you use olive oil to make soap versus using something like vegetable oil or lard, that's going to change your soap properties. The number of carbons in the chains also changes the properties, as does the number of double bonds. The next category of lipids includes phospholipids, and these are lipids that contain a phosphorus atom. We have different styles of them, and these are represented in the block diagrams. The first one here is our typical triacylglycerol, which contains glycerol plus 3 fatty acids. And if you insert a phosphate and an alcohol here, this makes it a phosphoacylglycerol. And then if your backbone, instead of being glycerol, is sphingosine, this is a sphingomyelin. So we've replaced glycerol with sphingosine, which has a long backbone that kind of wraps around. There's one fatty acid plus the phosphate and alcohol. So this is the structure of phosphoric acid, or H3PO4. And that can be used to link molecules together, which we'll see. If you have R groups on either side of that phosphate group, this is a phosphodiester. So in a C double bond O R group, that's just a regular ester. With a P double bond O to 2 OR groups, that's a phosphodiester. And the way that phosphodiesters appear in cells are as the minus charge here. So this is a phosphodiester in cells. Phosphoacylglycerols are also called phosphoglycerides. These are the most common type of phospholipid. So again, we have the block diagram with glycerol, the 2 fatty acids, a phosphate group connected to an alcohol, and this is what it would look like chemically. So glycerol has the 3 carbon chain that's shown in green there, and then we have 2 regular ester bonds. So there's 2 esters there, and then this is going to be our phosphodiester. So R double prime can be one of 2 different groups. If R double prime equals ethylamine, this is called a cephalin. Or phosphatidyl diethanolamine. Or if R double prime equals an ammonium salt, then this is called lecithin, or phosphatidylcholine. So just like in triacylglycerols, the 2 fatty side chains are nonpolar tails. The phosphoester end is charged, so that's kind of the polar head because it is charged. So we have a cephalin there because of that, the CH2CH2NH3, and that is derived from ethanolamine. And then the lecithin, like we mentioned, has this ammonium salt over here, and that comes from that choline alcohol. So our charged atoms are down here on the end. This is our polar head, and then we have 2 nonpolar tails. So these are often drawn as having one head there that's polar with the two tails. Another molecule that we discussed were sphingomyelins. These are the ones that don't have the glycerol backbone. These come from sphingosine, and that's the backbone. These do not have an ester because sphingosine is the molecule shown here, and this is the part that forms the bond. So this is an amide bond. All right, so you can see that right there highlighted in gray. So the amide bond forms with oleic acid that sphingosine wraps around. This is that part of the sphingosine. The general structure of the sphingomyelin has the sphingosine backbone, a fatty acid, which will form an amide bond. So this is an amide bond here. And then we have a phosphate alcohol, and here is a phosphodiester bond. And then depending on what your alcohol is on the phosphate, that will change the structure a little bit. So in this blue box here, you can have the choline or you can have the ethanolamine. Here we have a picture of different neurons. This is a neuron cell. And if we take a microscopic look at that, these kind of spread out and have different coatings. So there's layers of myelin sheath surrounding the nerve fibers, and we have myelinated axons as well. The myelin sheath coating that surrounds the nerve fibers also insulates the cells. People that have multiple sclerosis have scarring of the myelin sheath, or it's degenerating. Without the sheath, normal nerve transmissions are disrupted, can result in numbness and blindness, speech disorders, and also tremors. And inside of these myelin sheaths, we have the sphingosine, which you can see here. So that's the sphingosine part of the molecule. And that contains the nonpolar tails, and then you have the amide linkage here from the fatty acid. And then your choline, that forms a phosphodiester bond to the phosphate. And here is exposed nerve fibers. And that's damaged myelin sheath, which we see in multiple sclerosis patients. So just a summary here. A triacylglycerol has the 3 nonpolar side chains. Glycerol is the backbone. And it's got 3 ester bonds from the 3 fatty acids. A phosphoacylglycerol has 2 nonpolar side chain tails instead of 3 of the triacylglycerols, and 1 ionic head, so this is the polar head. There's 2 esters here and 2 fatty acids. This one also has the glycerol backbone. And the phosphate group is on the terminal part of the glycerol on the primary end. The sphingomyelin has 2 nonpolar side chain tails and 1 ionic head. So this is the polar part. The backbone is sphingosine. It contains an amide linkage. And the phosphodiester is located on the terminal carbon just like sphingosine. And this one's got 1 fatty acid. If we take a closer look at cell membranes, we have a barrier in the membrane that allows for ions to come in and out of the cell. This also allows the nutrients in and the waste to go out. Phospholipids, which we'll see, is a major part of these cell membranes, and it's made of a lipid bilayer, which is made from saturated fatty acid tails that pack together to make the wall more rigid, and these are the result of the nonpolar fatty acids. So as we've seen before, those fatty acid tails are made of a long chain of carbons, so that makes them have a nonpolar property, which we can see here. If you have saturated fatty acid tails, those pack well together and make the cell more rigid. And if you have unsaturated fatty acid tails, so there's carbon-carbon double bonds here, the wall is going to be more fluid. The head of the phospholipid is a polar head, because of that charged phosphate group and also that ammonium salt up there. So these are represented by the green balls, and you have this phospholipid bilayer that will accept ions in and things can go out as well. So the charged heads are on the outside, the tails are on the inside, and that serves as a barrier. And it's kind of like soap, except when we had the soap molecules, that formed a micelle or a circle around the grease bubbles. When phospholipids come in contact with water, they form a bilayer. There's different parts of the cell membrane structure. The little things that look like trees coming out there, those are carbohydrates. And these are known as peripheral proteins, so they're only on the outside. And these also happen to be glycolipids. So they're little carbohydrates that are attached to lipids that makes them the glycolipids. In this brown region over here where these tails come together to form that bilayer, that's the hydrophobic region. And that's a result of those saturated or unsaturated fatty acid tails. We can see cholesterol, the yellow blocks there. We have nonpolar regions, which come from the fatty acid tails, and then the polar regions are from the green heads. Other things that we have are integral proteins, which span the bilayer. So these extend the entire layer. And then we also have peripheral proteins. These are another kind. And those are just on one side or the other. So those carbohydrate ones are a peripheral protein, and then we have the ones at the bottom of those layers there. Within a cell, we have the cytoplasm, which is the watery stuff inside of the cell, and that is protecting the nucleus. And then we have that cell wall that is made up of that lipid bilayer. The lipid bilayer acts as a barrier, and it's going to allow certain things through. Oxygen and carbon dioxide are in high concentration on the outside. We have the larger polar molecules or ions cannot get through the bilayer. So it's not easy for them to go across. Things like Cl- or HCO3- will have a hard time crossing the border. And so we have different types of transport here. On the inside of the cell membrane, our O2 and CO2 is in low concentrations. So of the different types of transport across the cells, there's simple diffusion, where the molecule can go through pretty easily. We have facilitated transport that comes through an integral membrane protein. So if the chlorine ions wanted to come into the cell, that's going to require some help from the integral protein, so that one won't transport easily. Oxygen And carbon dioxide can go through easy with the simple diffusion, whereas our ions and larger polar molecules are going to need some facilitated transport through this integral membrane protein. And then there's also something called active transport. And if you're dealing with active transport, energy input is needed because usually something is going against the gradient. So in this one, we have our sodium, potassium, and calcium cations are moving against the gradient. So it goes from areas of low concentration to high. And so it needs something to help it get through the channels. So this happens when the nerve causes muscles to contract. We're going to move against the gradient, so that's going to require an escort. So active energy is needed to get those ions out.